At first glance, the slow loris seems almost designed to be underestimated: huge eyes, rounded face, quiet posture, and movements so careful they can look sleepy. But this first impression is deceptive. Slow lorises are not passive mascots of the rainforest. They are highly specialized nocturnal primates with a survival strategy built around stealth, grip control, and chemical defense. They belong to the genus Nycticebus and occur across a broad arc of South and Southeast Asia, from parts of Bangladesh and Northeast India through mainland and island forests, down to Java, and eastward toward the Sulu region. They are difficult to study in the wild because they are active at night, often silent, and typically hidden in dense canopy structure. That lack of visibility has helped myths spread faster than biology. To understand slow lorises properly, we have to strip away the viral image version and replace it with an ecological one: an animal shaped by darkness, predation risk, territorial pressure, and shrinking forest networks. One reason slow lorises are often misunderstood is that even scientists have revised how they classify them. Earlier systems sometimes grouped many populations into very few species, occasionally treating them as one broad species with local variants. As researchers compared skull shape, facial patterns, body proportions, geography, and genetic signals in more detail, that broad grouping became harder to defend. Today, multiple species are recognized within Nycticebus, including Sunda, Bengal, Javan, Philippine, Bangka, Bornean, Kayan River, and Sumatran forms. In addition, the pygmy slow loris has been moved into a separate genus, Xanthonycticebus. This shift is not just academic housekeeping. Classification directly affects conservation outcomes. If many isolated populations are merged under one label, severe local declines can be hidden inside a larger range map. But when populations are recognized as distinct species, their restricted distributions and unique threats become visible, and protection can be designed with more precision. In conservation, names are not cosmetic. Names determine who gets counted before they disappear. Their place in the primate family tree adds another layer of importance. Slow lorises are closely related to slender lorises of southern India and Sri Lanka, and more distantly related to African lorisids such as potos and enguantabos. Farther away are galagos, and farther still are lemurs in Madagascar. This branching pattern matters because it reminds us that primate evolution is not a single track toward one body plan. Different lineages solve different ecological problems. Slow lorises represent one of the most unusual solutions: a nocturnal, branch-dependent primate that emphasizes precision over speed and includes a rare mammalian toxin system. Their fossil record is limited, and molecular timing studies have produced inconsistent dates, so parts of their deep history remain unresolved. But uncertainty about exact dates does not weaken the core insight. Slow lorises are evolutionarily distinctive, and they preserve a set of traits that are uncommon among primates. Losing them would not just reduce animal numbers. It would erase a unique evolutionary strategy from living ecosystems. Now look at the body design itself. Slow lorises have large forward-facing eyes suited to low-light vision, a relatively narrow snout, and a long, flexible torso. Their limbs are close in length, and their hands and feet are adapted for prolonged, forceful grasping. This gripping system lets them hold branches for extended periods with stability that supports their deliberate movement style. In the canopy at night, this matters enormously. Fast movement can create noise, vibration, and mistakes. A slip can mean injury or exposure. Slow lorises reduce those risks by moving in controlled sequences: reach, test, shift weight, secure contact, then advance. It is less dramatic than leaping primates, but for their ecological niche it is highly effective. Their morphology is not primitive. Leftovers from an ancestral form. It is a tuned structure for arboreal caution under low visibility. What appears clumsy by daytime human standards is actually a precision mode that prioritizes safety, silence, and positional control in a three-dimensional environment full of hidden danger. The most extraordinary trait of slow lorises is their toxic bite, a feature that is rare in mammals and unique among primates. The mechanism involves a secretion from a gland on the arm. The loris licks this gland, mixing the secretion with saliva, and then delivers it through a bite. Earlier interpretations emphasized predator deterrence, and that role likely exists. But evidence increasingly suggests the toxin system is also important in conflicts between lorises, especially territorial disputes. That changes the story from occasional emergency defense to regular social weaponry. If a trait is used in intraspecies competition, it can influence spacing patterns, mating dynamics, injury rates, and dominance boundaries. Chemically, the secretion includes compounds related to known allergenic proteins, and wild diet may contribute additional toxic complexity. In other words, this is not a cartoon venom switch. It is a layered biochemical system embedded in behavior and ecology. The slow loris is therefore not just a primate with a dangerous bite. It is a primate whose social and defensive life is partly mediated by chemistry. Their threat response reinforces this strategy. Many animals respond to danger by fleeing fast, vocalizing loudly, or displaying obvious warning postures. Slow lorises often do the opposite, they freeze. They remain still, silent, and visually inconspicuous, sometimes for extended periods. In dense nocturnal environments, stillness can be protection. Movement attracts attention, immobility can break detection. Documented predators beyond humans include snakes, changeable hawk eagles, and even orangutans, while additional predators are suspected in parts of their range. Against that mix, a low-profile approach makes sense. Their survival stack works in layers: avoid detection through quiet movement, avoid escalation through freezing, maintain branch security through strong grip mechanics, and keep toxic bite capability as a close-range option. This is why calling them slow in a dismissive sense misses the point. Their speed profile is not failure. It is risk management. They sacrifice travel pace to gain stealth, control, and tactical flexibility in a habitat where one panic decision can be fatal. Social behavior in slow lorises is less visible than in many daytime primates, but chemical communication appears central. Scent marking allows individuals to project information through time, not just in the moment. In dark forests where visual signals are limited, scent can encode territory, identity, and reproductive context in ways that persist after the animal leaves. Males are often described as strongly territorial, which aligns with evidence for biting in social conflict. Territory here is not abstract space. It is access to food trees, gum sources, safe routes, and mating opportunities. Reproduction is slow, and infants may be parked on branches or carried by either parent, highlighting both parental investment and vulnerability. This life history pattern has major implications. Species with slow reproduction cannot quickly replace adults removed by hunting or trade. So even moderate annual losses can trigger long-term decline. In practical terms, slow loris populations are not built for repeated extraction. Their social system and reproductive tempo evolve for ecological stability, not for resilience under rapid human pressure. Diet deepens the picture. Slow lorises are omnivorous, feeding on small animals, fruit, tree gum, and other plant material. That sounds flexible, but ecological flexibility is not the same as habitat independence. Tree gum feeding, for example, depends on suitable vegetation structure and repeated access to known feeding points. Fragmented landscapes disrupt those pathways. A canopy-adapted animal forced into open gaps faces exposure, stress, and predation risk. Diet may also interact with toxin biology. Some researchers suggest wild food intake could influence secondary chemical compounds associated with their defensive system. If true, habitat degradation does more than reduce shelter and food quantity. It may alter the chemical context that supports defensive function. This means conservation cannot focus only on preserving tree cover as a percentage. It must preserve functional forest, the right mix of species, canopy continuity, and foraging opportunities across time. Otherwise, lorises may remain present on paper while losing the ecological conditions that make long-term survival possible. Conservation assessments already signal serious risk. Species recognized before recent taxonomic revisions have often been listed as vulnerable or endangered, and newly distinguished species are expected to face at least comparable pressure, sometimes greater because their true ranges are narrower than once assumed. Two drivers dominate: habitat loss and wildlife trade. Forests are cleared, simplified, or split into fragments that isolate groups with limited dispersal ability. At the same time, unsustainable capture for exotic pet markets and traditional medicine removes individuals directly from those shrinking populations. The interaction is destructive. Fragmentation increases vulnerability to capture, and capture weakens already fragmented populations by removing breeding adults. Because slow lorises reproduce slowly, population recovery lags far behind extraction speed. This creates a dangerous illusion: numbers can decline quietly for years before collapse becomes obvious. By the time the problem is visually undeniable, many local systems may already be below recovery thresholds. Effective action has to be early, targeted, and persistent. Public perception is a critical battlefield in this story. Animals with expressive faces are often treated as emotionally familiar, and that familiarity can create false confidence about captivity or handling. Slow lorises are especially vulnerable to this bias. Their appearance invites anthropomorphic interpretation, while their biology strongly warns against domestication and trade. They are nocturnal specialists with complex ecological and social needs, and they possess a toxic bite system that itself reflects how unsuitable close human interaction can be. A key challenge in conservation communication is replacing fascination-driven demand with respect-driven distance. People do not need to fear slow lorises, but they do need to understand them accurately. Educational framing that emphasizes uniqueness without romanticizing ownership can reduce harmful demand. In policy terms, messaging is not secondary to enforcement. It is part of enforcement. If cultural narratives continue to normalize possession, supply chains regenerate despite legal pressure. If narratives shift toward ecological responsibility, demand can decline and legal tools become more effective. What would serious protection look like? First, species-level planning is essential, because each species faces different local threats, governance conditions, and habitat patterns. Second, habitat strategy must prioritize connectivity, not only isolated protected patches. Gene flow and movement routes are critical for long-term viability. Third, anti-trafficking work must combine enforcement with demand reduction, targeting both capture networks and consumer behavior. Fourth, research gaps need sustained support, better distribution mapping, population trend monitoring, reproductive ecology, and toxin-related field studies are all practical management tools, not abstract academic goals. Fifth, local communities must be integrated into conservation design through incentives and co-managed approaches that align protection with livelihoods. Without local legitimacy, external conservation plans often fail after initial funding cycles. The broader lesson is that no single intervention can secure slow lorises. Their future depends on coordinated systems thinking, taxonomy, habitat science, law, behavior change, and community governance working in parallel over long timelines. In the end, slow lorises challenge two assumptions at once. Biologically, they challenge the idea that successful primates must be fast, loud, or socially conspicuous. Their strategy is almost the opposite: deliberate movement, sensory subtlety, and chemical escalation when necessary. Ethically, they challenge how humans respond to rarity and charisma. The same traits that make them memorable can increase pressure through demand, curiosity, and exploitation. If we keep seeing slow lorises as novelty animals, we accelerate their decline. If we recognize them as evolutionarily distinct forest specialists under compound threat, then attention can become protection instead of extraction. Their story is not just about a toxic bite. It is about whether modern conservation can act quickly enough for species that recover slowly. A slow loris can survive by moving carefully in the dark. Our challenge is different, to move decisively in the light, before quiet losses become permanent ones. If you found this helpful, please like this video and subscribe for more explainers.